Structure of blood. The cardiovascular system circulates blood which carries nutrient, waste products, hormones, and other important substances throughout the body. Plasma. Plasma, also called blood plasma, is the liquid part of the blood. Plasma is a yellow fluid composed mostly of water. Elements suspended or dissolved in the plasma include gases, electrolytes, carbohydrates, fats, proteins, clotting factors, and waste products. Helpful hint. Whole blood is a blood specimen with an anticoagulant that has not been centrifuged. The component must be gently mixed before testing can be done. The cells suspended in plasma are the formed elements. Red blood cells transport oxygen throughout the body. Red blood cells contain hemoglobin, a large molecule with iron atoms that bind to oxygen. White blood cells fight infections. Platelets, also called thrombocytes, gather at sites of damage in blood vessels as part of the blood clotting process. Did you know that thrombocytopenia is an abnormally low level of platelets? To collect plasma, the blood specimen must be treated with an anticoagulant. Blood specimens with an added anticoagulant are called whole blood specimens. When centrifuged, whole blood specimens will separate into 3 layers: plasma, white blood cells, and platelets, buffycoat, and red blood cells. The next one is serum. Serum is plasma with clotting factors, fibrinogen removed. So serum is plasma with clotting factors removed. Serum is collected by allowing a blood specimen to coagulate. After centrifugation, a serum tube will have 2 layers: serum and clotted blood. In a serum separator tube, SST, a gel layer will separate the serum from the clotted blood. Blood groups, also called blood types, are determined by the presence of antigens, proteins that activate antibodies. Antibodies are immune system proteins that bind to antigens to destroy the attached cell or pathogen. Each blood group produces specific antibodies that attach to antigens on RBCs. When antibodies attack RBCs, the cells will agglutinate, clump, and lyse, which is LYSE, break apart. A process called a transfusion reaction. When receiving a blood transfusion, a person should receive their own blood type to avoid transfusion reaction. The ABO blood group is defined by the presence or absence of A antigens and B antigens. A person's RBC may contain A antigens, B antigens, both antigens, or neither antigens. If a person does not have one type of antigen, their plasma will contain antibodies that attack that antigen. Example: a person with type A blood will have antibodies that attack type B blood cells. So a person that have O blood type, antigens and RBC on their red blood cells is none. The antibodies in their plasma will be antigen A and antigen B antibodies. A person that has AB blood type will have A and B antigens. The antibodies in the plasma will be no antibodies. Person that has blood type A will have A antigen on their red blood cells and antigen B antibodies. And a person with blood type B will have B antigen and antibodies and antibody A antibodies. The Rh blood group is defined by the presence of Rh factor and an antigen, also called D antigen. The blood type Rh positive has Rh factor antigen on their red blood cells. The blood type Rh negative does not have Rh factor antigens on red blood cells. Anti-Rh antibodies develop when Rh negative blood is exposed to Rh factor. In hemolytic disease of newborn, anti-Rh antibodies in an Rh negative mother attack the RBCs of her Rh positive fetus. Did you know pregnant people who are Rh negative receive a shot that prevents their body from producing anti-Rh antibodies? This protects the fetus from hemolytic disease of the newborn. Quick review question. What function do red blood cells perform in the body? Why is an anticoagulant needed to collect a plasma sample for diagnostic testing?